Good day, everyone. I am Sir Franz, and I'd like to welcome you to our lesson on the physiology of the liver. And I'd like to encourage you as you continue to have your internship and as you continue to review for your physician's licensure examination. So, this will be the topics for discussion at this time. You have to understand that the liver is an accessory structure of the gastrointestinal tract. And so the function of the liver is intimately tied to that of the alimentary tract or the digestive tract. The liver is the first site of processing of most absorbed substances, whether those are foodstuffs or toxins. It's the liver that first comes in contact with those that we have absorbed. And the liver secretes bile, which is critical for lipid absorption. Bile, if you think about it, is technically soap. And so the fat that is in the food mixes with the bile, the soap, so that the lipid can emulsify into the water of the digestive fluids or the chyme. The liver handles xenobiotics. Xenobiotics, ibig sabihin yan, any foreign substance relative to the endogenously produced substances of the body. The liver handles xenobiotics and metabolizes them into excretable forms. And in toto, the goal of biotransformation would be to make a lipid-soluble poison or substance into something that is more water-soluble so that it can freely undergo glomerular filtration and um, is hard to reabsorb because it is already polar. First point of discussion would be the metabolic functions of the liver. The liver engages in gluconeogenesis, which is the making of glucose from non-carbohydrate sources. And the liver also functions in glycogen metabolism, whether that is glycogen catabolism, glycogenolysis, or glycogen synthesis, which is glycogenesis. So, because of this function of the liver is touted to be the glucose buffer of the body. So, um, think of it this way na kung meron kayong kinain, you just digested it, made it something more simple, you absorbed it, the liver will trap the glucose so that postprandially there is no severe hyperglycemia. So the liver traps the glucose by attaching a phosphate group into it and therefore traps it intracellularly of the hepatocyte. So um, the venous outflow from the liver already has little glucose kasi na-trap niya na. So yun kapag meron kang kinain. However, when you don't have something to eat, when you are fasting, the liver does the opposite. It supplies glucose back into the blood via gluconeogenesis and via glycogenolysis. And so, na-fulfill ng liver ang kanyang buffer function. Kung mayroong sobrang glucose, ipe-prevent niya na sobrang dami yung nasa dugo. Pag sobrang konti naman yung glucose na nasa enteric tract, then it will also prevent a fall in blood glucose. So that is the principal glucose-related problem of patients with end-stage liver disease. there is an erratic excursion of glucose, no? Pag mukaon sila, the glucose shoots up, and then um, when just a little bit of time, when they don't have something to eat, they're fasting, they tend to experience hypoglycemia. So there are periods of hyper and hypoglycemia. The liver is rich in enzymes for fatty acid oxidation. So enzymes for lipolysis. And so if you will remember your biochemistry, the free fatty acids that have been released from the adipocytes by the action of katong hormone-sensitive lipase activated by epinephrine um, will cause an increase in free fatty acids in the blood. So the free fatty acids then go into the liver where beta oxidation occurs, and the excess acetyl coenzyme A floods the Krebs cycle. So kapag ma-flood na ang Krebs cycle, kaya dagan na kay siya acetyl coenzyme A, the excess acetyl coenzyme A is fused to form ketone bodies. This time, the ketone bodies are a much well-accepted source of energy in the form of fat. So technically, the liver functions there as an intermediary processor para magamit sa other parts of the body ang fat uh, through the production of ketone bodies. On the opposite end of the spectrum, if there are excess products of carbohydrate metabolism, the liver converts it into free fatty acids and lipids, particularly triglycerides, which exit the liver via VLDL. And the VLDL will then transport those triglycerides into the periphery, where the enzyme that is in the endothelium, katong lipoprotein lipase, will cleave off the triglycerides and release the free fatty acids, which are then taken up by the adipose. So dito niya ma-store sa sulod sa adipose in the form of a triglyceride droplet. And uniquely, the liver functions as some sort of cholesterol control mechanism, no? Ang excess cholesterol, and in particular, majority of the cholesterol in the plasma is converted by the liver into bile acids. And because bile acids are excreted into the enteric tract where they can be not reabsorbed, that pathway is a pathway that eliminates cholesterol from the body. Okay? And so, doon siya magiging unique, no? Yung understanding nyo of the cholesterol physiology, bile acid physiology. Kasi if you chelate the bile acids in the enteric nervous system, then the liver will have to be forced to convert a larger proportion of the serum cholesterol into bile acids kasi walang bumabalik sa kanya via enterohepatic recycling. Okay, so this is a nice image from Byrne and Levi. You have here the portal vein, which is the confluent venous drainage of the gastrointestinal tract. And all of it will have to pass through the liver. And for example, in the fed state, ito magiging rich in glucose. This will travel the portal vein, and once it encounters the hepatocyte plates in the liver, those hepatocytes will then take up the glucose. So para ding yung portion of glycolysis, and then convert the glucose into glucose 6-phosphate. Once intracellularly, the glucose 6-phosphate is already present because of that phosphate group, which makes the glucose relatively very polar. It cannot cross back to the cell membrane and therefore is effectively trapped inside the hepatocyte. So, kukonti na ngayon yung lalabas na glucose through the vena cava and into the circulation. So that is a glucose buffer kung sobrang dami nung carbohydrate in your entire tract. But on the opposite end of the spectrum, if kumukonte na yung na-absorb yung glucose dito, the liver will then start to release glucose via glycogenolysis and gluconeogenesis so that the levels of glucose in the systemic circulation is maintained. So buffer pa rin talaga siya. The liver, synthesizes non-essential amino acids. So when we talk of the non-essential amino acids, these are amino acids that do not need to be repleted through the diet. Okay, so the body has a way of synthesizing these amino acids from the scraps of other materials found inside the human body. So it relies on isomerisms and transaminations, which is a major role of the liver or the hepatocyte. Yan. So the liver interconverts and deaminates, tatanggalan niya ng amino group, yung mga amino acids, so that the alpha keto acid or the carbon skeleton of the amino acid remains. And so that carbon skeleton of the amino acid, the alpha keto acid portion, will be used as a substrate for gluconeogenesis. So via this pathway, the hepatocyte enables the body to produce glucose from amino acids. So gluconeogenesis. But of course, hindi naman part ng glucose yung amino group, kailangan mo yung tanggalin via oxidative deamination and transamination. The liver synthesizes almost all of the plasma proteins. So, more importantly, it synthesizes albumin, which determines the plasma oncotic pressure and holds the water inside the intravascular space. Albumin is also important because it is a marker of nutrition. So whenever the plasma albumin drops below 3.5 grams, that is already an indicator that the patient may have chronic malnutrition. Okay? Kasi kailangan mo naman talaga ng food intake to sustain the amino acid requirements to make albumin. And so patients who have, for example, kwashiorkor, who are deprived of protein, they are not able to synthesize albumin, and so they will have anasarca or generalized edema. The liver also synthesizes the clotting factors, which are also again proteins, but again are important for the coagulation cascade. So hindi na katakataka that if you have liver injury or end-stage liver failure, cirrhosis, these patients tend to bleed a lot because the mass, the hepatic mass has decreased so much that there is no synthesis of clotting factors. And via the action of interleukin 6, the liver synthesizes the acute phase reactants, which are collectively called the pentraxins. Kasama dyan yung mga complement proteins at saka yung C-reactive protein. Those are synthesized by the liver in response to an inflammatory mediator, which is interleukin 6. And so, isa pang clinical application, the immunophenotype of a patient who has end-stage liver disease can be erratic. Pwede na meron silang severe inflammation or infection ongoing pero wala kang makikitang increase in C-reactive protein kasi wala ngang function yung hepatocyte in synthesizing the pentraxins. And again, kanina sabi natin, yung liver or hepatocyte, it deaminates amino acids. And so that ammonia is toxic. So kailangan mong i-chelate technically yung ammonia with carbon to form urea so that it is eliminated by the kidneys. And when this ammonia is not effectively handled by the liver, for example, in patients who have hepatic encephalopathy or hepatic coma, yung ammonia na yon hindi nako-convert to urea, the ammonia accumulates and is circulated into the brain, passes the blood-brain barrier kasi it's relatively non-polar. So yung ammonia will then attach itself, no, or incorporate itself into the alpha keto acids that are found as intermediates of the Krebs cycle. So technically kinakain ng ammonia yung intermediates of the Krebs cycle, and so the neuron of the brain will not be able to produce ATP, producing your encephalopathy. So next function, the liver and detoxification. This is a nice analogy by si Byrne and Levi na sinabi niya that the liver does this function via two ways. First would be the gatekeeper function. The liver limits the entry of toxic substances absorbed from the gastrointestinal tract into the bloodstream. So, kaya dito man siya una mo flow ang blood, then pag if you have ingested the poison, then that first encounter by the liver already eliminates the toxic substances absorbed per orem, delivered per orem, sorry. Then it has this second function, the garbage disposal function. It extracts potentially toxic metabolites and converts them to chemical forms that can be easily excreted. And these are more polar, more water-soluble forms. Please take note of this phenomenon, the first pass effect or first pass metabolism. It describes the instance where substances absorbed through the gastrointestinal tract do not reach the systemic circulation, as they are eliminated by passing through the liver. So, notorious for this would be some medications in your pharmacology na kung mapapansin nyo, sobrang taas ng dose. For example, the nitrates, for example, beta blockers, for example, opioids. Relatively, compared to other therapeutic classifications of drugs, matataas yung dose nila. They are less potent. And that is because the dose is it is planned to be high so that after first pass metabolism, meron pa ring matitira that provides the therapeutic levels in the plasma. No? You have to, the dosing, the posology would have to incorporate the first pass effect. Kaya mataas yung dose. So first would be physical extraction by Kupffer cells. Ito mga Kupffer cells, these are specialized macrophages inside the liver. And if you will recall your histology, itong mga Kupffer cells, they have been in the liver since the development of the liver. So hindi sila galing sa bloodstream and then hones into the liver, no? Homes into the liver, hindi sila ganon. Much like the macrophages come from monocytes in the blood. Ang Kupffer cells, pagka produce pa lang ng liver bud, nandun na sila. Okay, so they are native macrophages of the liver. Blood arriving in the liver will have to percolate along the sinusoids, and we'll have to encounter the Kupffer cells which lines the endothelium. And so the particulate material and bacteria are phagocytosed. So this is important kasi although classically described yung enteric epithelia as having tight junctions, there are kumbaga parang misconceptions pa, no? They're not really that tight. And so some particulates from the foodstuff and even whole bacteria can go into the portal venous drainage and go into the liver. But have you ever wondered bakit hindi tayo nagsisepsis? Because majority of those particulate material and whole bacteria are phagocytosed by Kupffer cells. Then the next, kumbaga, line of defense by the liver in detoxification would be metabolism by hepatic enzyme systems. So the hepatocytes have a lot of smooth endoplasmic reticulum. And so itong smooth endoplasmic reticulum, the primary goal of this would be lipid metabolism. So the enzymes that perform drug metabolism or synobiotic metabolism are also found in the smooth endoplasmic reticulum. So these enzymes will perform chemical reactions with the goal of making a synobiotic or poison more water soluble. So this let's orient. Ito yung sinusoidal lumen. So this would be the sinusoidal capillary, no? Na produced by the fusion of microscopic branches ng portal vein at saka ng hepatic artery. So that makes the sinusoid, blood will flow here, and this would be the endothelium of the sinusoid. So maraming fenestrations yan, di ba? So papasok siya dito, yung kung ano-anong substances na yan, plasma, what else. And then that's the time that it bathes the hepatocyte. Okay? So for example, if you have a poison, let's say, meron tayong example mamaya, no? Paracetamol. Punta siya dito, the hepatocyte will then actively take up the paracetamol, and it finally finds its way into the smooth endoplasmic reticulum, where the cytochrome P450 and the conjugating enzymes are present, which effectively detoxifies the paracetamol. And then yung processed na paracetamol na less ano na siya, less lipid soluble, so it is now more water soluble, would be actively pumped into the bile canaliculus and goes into the bile. So pagpunta niya sa bile, kagaya din nung biliary cycle natin, maka-absorb din yung gamot na yun, but this time because it's more water soluble, after entering the systemic circulation, some of it will find its way into the kidney where glomerular filtration occurs. And maiihi mo na siya. And then, in the case of physical kumbaga detoxification or cleansing by the liver, if, for example, you have a whole bacteria here, kakainin lang siya ng Kupffer cells. So that walang makakapunta, no? Papunta sa hepatic vein and pupunta sa systemic circulation. So let's dig deeper into the synobiotic metabolism. You have 4 classically described reactions for phase 1. oxidation. And when there is oxidation, there's also reduction. You take out an electron, you add an electron. Hydroxylation, you add an OH. And hydrolysis, you break an ester by adding H and OH. So these reactions are mediated by the cytochrome P450 family of enzymes. So please remember, cytochrome P450 family of enzymes. So itong mga enzymes na ito, they also contain a lot of ions such as iron and copper. So um, this same cytochrome P450, some of the uh, isoforms of this would be found in the mitochondria, and those isoforms of cytochrome facilitate electron transport chain kasi meron ding kumbaga ping-pong mechanism with the ions of iron and copper in those cytochromes. But this time, this isoform is found in the smooth endoplasmic reticulum and it catalyzes these reactions. What is the goal of phase 1? The goal of phase 1 would be to attach a polar handle. Magdadagdag siya ng isang moiety into the drug, and that moiety is very polar. And that moiety is, aside from being polar, is highly reactive. So the goal of phase 1 would be to attach the handle so that that handle will be utilized by the conjugating enzymes, which is already phase 2. So phase 2 reactions or conjugation reactions mean that there is an attachment of a big polar molecule to that polar handle na ginawa ng phase 1 to drastically increase the water solubility of the xenobiotic. So for example, you have glucuronic acid. So ang structure nito would be, you have glucose, and then on carbon number 6 of glucose, di ba meron yung hydroxyl group? I-oxidize mo yung hydroxyl group into a carboxylic acid, and that will be glucuronic acid. So, dahil sa dami ng hydroxyl groups ng glucose, pag inattach mo yon sa isang xenobiotic, the xenobiotic becomes effectively more water soluble. Another one would be sulfate conjugation. You have amino acid conjugations, common dito si glycine, and glutathione conjugation, which is made up of three amino acids. No? It is a tripeptide, yung glutathione. Glutamate, glycine, and cysteine. So, yun yung tatlong amino acids in glutathione. Okay? So, of course, the norm is magfe-phase 1 muna yung xenobiotic, and then after phase 1, pupunta siya sa phase 2. You might also be aware that the enzymes of phase 1 reaction are inducible. Ibig sabihin, there are certain conditions or certain substances that will increase the levels of the cytochrome P450 family. And that means, yung mga gamot or substances that are metabolized via CYP450 would be metabolized faster. And so you will have a lower therapeutic concentration in the blood. And then there are also inhibitors which will do the opposite. Okay? So, next. This would be the best example. This can also be discussed in your pathology or emergency medicine. You have paracetamol. So, paracetamol, ang pangalan niya because it is made up of a para acetaminophenol. So you have your phenol group, and then para position to that, 1, 2, 3, 4. Then you have the amine group. So technically para siyang parang aniline din. So meron kang amino group here, and then merong acetate, carbon 1, carbon 2. So yan ang structure ng paracetamol. The majority of the paracetamol will be catered by the phase 2 reaction of glucuronidation, where dito sa hydroxyl group ng paracetamol, merong ma-attach na glucuronic acid. Here's this one. This will be very water-soluble already. However, if you have an overdose, na-saturate mo lahat ng enzymes, the UDP, glucuronosyl transferase, na puno mo yon kasi ang dami ng paracetamol na ininom, the remainder of the paracetamol will be catered by sulfation or addition of sulfate group, this one. If napuno mo pa talaga kasi sobrang dami ng paracetamol, the paracetamol will then be shunted away from the conjugating pathways and into a CYP predominant metabolic pathway. But this time, the paracetamol becomes oxidized. So, mapapansin nyo, nag-develop na siya ng double bond at saka ng ketone group. So, this is the very reactive species NAPQI, N-acetyl paraquinone imine. So, ito yung quinone group. So, pagdating dito, it's not yet the end of the world. Pwede pa siyang dikitan ng glutathione. Okay? So, that's your glutathione conjugation. However, if you still saturate this pathway, NAPQ will accumulate and this will form toxic adducts with your proteins and nucleic acids that will lead to necrosis of hepatocytes. In the Western countries, ang inyong paracetamol overdose is the leading cause of acute liver failure requiring liver transplantation. So, dito din pumapasok, by knowing this pathway, dito pumapasok yung paggamit natin ng N-acetylcysteine or fluimucil ang brand as an antidote to paracetamol poisoning. Kasi yung cysteine component ng N-acetylcysteine will provide the necessary amino acid, which is cysteine, to make more glutathione para mas ma-empower mo tong pathway na to and ma-cater mo yung accumulating NAPQ. Next, aside from the function, let's go a little bit into physiologic anatomy of the liver and the biliary system. So hepatocytes are arranged in anastomosing cords, but in pathology we usually call it plates, around which large volumes of blood circulate or flow through. So kung ano man yung substances na nasa blood, the liver will be actively taking some of it, no? Whether those are nutrients, those are ions, proteins, kukunin yan ng liver. Okay? So, please take note of this. Majority of the blood that goes into the liver comes from the portal vein. But by volume, by concentration, no? Volume per volume concentration, the amount of oxygen would be lower in portal venous blood. So, mas maraming portal venous blood na pumupunta sa liver, and that is the major blood supply of the liver, but the richness of the oxygen is lower in the portal vein. Of course, mas marami yung galing sa hepatic artery. Take note of, wag niyo take note, but just appreciate na from 70% na wala kang kinakain, kapag kumain ka na, it rises to 90% of the total blood flow, no? From the portal vein. And of course, ano naman siya, no? Parang logical naman siya kasi the control of blood flow into the gastrointestinal tract, stomach, small and large intestines, is dependent on the activity of the epithelia. So, kung mas muli, pag mas musecrete ang epithelia, mas kailangan niya og ATP, mas kailangan niya dagan na blood flow. Pag mag-produce siya og peristalsis, mas kailangan niya dagan blood flow. So all of those blood that goes into the gastrointestinal tract by way of the celiac, SMA, IMA, of course, mubalik to siya back into the systemic circulation. But of course, it has to pass through the portal vein. No? So, kaya tataas after the postprandial state. Sinusoids are low resistance capillaries supplied by microscopic branches of the portal vein and hepatic artery. So, during fasting, this is amazing. Kaka-learn ko lang din ito. Many sinusoids are collapsed, but as the blood flow into the gastrointestinal tract increases, portal venous blood flow also increases, more and more sinusoids get recruited. From the liver, blood drains into the hepatic vein. The space of this, which is the space between the liver plate and ang katong endothelial cell of the sinusoid, it holds Kupffer cells and stellate cells. So stellate cells are, you can think of it as a fibroblast. No? It's a fibroblast, it's a phagocytic cell, but at the same time, it also stores vitamin A, parang ETO cells. No? It pathologically synthesizes collagen when it is activated by dysregulated inflammation. Okay, and so the stellate cells would be the source of collagen for fibrosis during liver injury or cirrhosis. So, di ba, yung staging natin ng cirrhosis depend on the state of fibrosis. Now, we know that that fibrous tissue that forms the regenerative nodules come from the stellate cells. The hepatic vein, hepatic artery, and bile ducts, they make the hepatic triad. And based on the hepatic triad, we have zone 1, 2, and 3. Zone 1 cells lie far from the hepatic vein. So kung asa nagsugod ang sinusoid, naa dito ang zone 1 hepatocytes. They receive the most oxygen and nutrients kasi sila man ang first contact. They are more active in detoxification and are thus susceptible to oxidative injury. Kaya daghan mag-oxygen dito, so mas daghan og oxygen radicals na ma-form dito. Zone 3 cells lie near the hepatic vein kung asa mag-drain. And so, dahil layo siya sa hepatic, layo siya sa, dahil malayo na siya sa start, zone 3 cells have low oxygen. But they are thought to be the most active at bile synthesis. But, kasi wala na siya masyado oxygen, they are most susceptible to ischemia. So, focus muna tayo sa B, no? Ito yung hepatic lobule. It classically forms a, unsa man siya, hexagon. It forms a hexagon with the central vein in the middle. Mo siyang central. And this is your portal triad. Microscopic branch of the portal vein, artery, and bile duct. So, remember that the portal vein and the hepatic artery will contribute its endothelium to form the sinusoid. So blood will start here and go towards the middle. So nandito yung zone 1, nandito yung zone 3, nandito yung zone 2, hepatocytes. So let's now focus on A. Venous blood from the intestines go through here and it passes through the sinusoid. So the sinusoids technically are capillaries, dadaan ng dugo dyan, and the plasma will exude into the space of this and it will bathe the hepatocyte. So kung sa man itong mga kuwaon sa hepatocyte, makuha na niya. No? And kung sa man itong ilabay sa hepatocyte, malabay na niya through bulk flow. So it starts here and ends at the central vein where systemic circulation is re-entered. So moto siyang blood flow, but uniquely, the apical cells, the apical membrane of the hepatocytes, they fuse together to form the bile canaliculus. So kani siya, ang classic description ani niya would be anti-parallel flow. From portal vein to the heart, to the central vein, in that direction, that's the flow of blood. But the flow of bile will be anti-parallel from the central vein back into the portal triad, where it empties into the bile duct or the canals of Herring. Yeah. So we have discussed that, and so let's discuss the mechanism of ascites. If the sinusoids are compressed by fibrosis, it loses its ability to accommodate. Kanina sabi natin, kapag kumain na siya, ang isang person, because low resistance ang sinusoids, the increase in flow will cause recruitment of the sinusoids. But if the sinusoids are compressed by fibrosis, remember the stellate cells are in the space of this. And so when they deposit the collagen, eventually it compresses the sinusoid. Okay? So pag inipit niya na yung sinusoid, the postprandial increase in blood flow will be met with an increase in pressure because your originally low resistance sinusoid becomes a high resistance vessel. Due to fenestrations in the sinusoid, the sinusoidal endothelium, albumin escapes from the intravascular space and an albumin-rich fluid will weep from the surface of the liver, so magsingot ang liver, into the abdominal cavity where it overwhelms the lymphatic drainage of the parietal peritoneum. So here would be a picture from Dr. Netter saying to us the pathophysiologic factors in ascites. Number one would be portal hypertension. And why is there high pressure here? Because the sinusoidal capillaries inside are being squeezed by fibrosis. Next, liver damage. So you have to understand that majority of the blood flow that goes into the liver comes from the portal vein. And so if the sinusoids are repelling blood flow into the liver, then the hepatocytes will chronically be exposed to ischemia. Chronic ischemia siya. And so mamamatay yung liver cell. That's one mechanism. But the other mechanisms of liver damage are from the etiology of the cirrhosis. But at the end of the day, the liver loses its function. So, hindi na siya makakagawa ng plasma proteins, hypoproteinemia. When there's hypoproteinemia, the oncotic pressure inside the blood vessels, they will decrease. And so, water will tend to escape the intravascular space. Next, because water tends to escape the intravascular space, the intravascular volume collapses. And so that will be detected by the macula densa of the kidney as if kulang ng dugo. So i-activate niya ngayon yung RAAS. So with the activation of the RAAS, um, you will be reabsorbing a lot of sodium and water. But again, that reabsorbed sodium and water loses its ability to stay inside the intravascular space. And so it becomes a vicious cycle. No? Kaya and take note, this letter B is the most important mechanism in ascites formation. So that is why the cornerstone in treatment of ascites would be fluid and salt restriction. Okay? Plus use of diuretics. Kay imo mang dapat tanggalon ang asin, which is pathologically increased. So next, there would be lymphatic changes. And so the lymphatic system is not able to reabsorb the fluid that exudes from the sinusoidal capillaries. And so they trickle from the surface of the liver. And the next, in this in portal hypertension and in ascites, there is a preponderance of release of vasodilatory substances, no? So that will cause increased capillary permeability systemically. So that aside from accumulation of fluid here in the peritoneal cavity, mag-accumulate din ng fluid sa dependent portions of the body. Kasi systemic man ang increase in capillary permeability. Next, let's discuss bile physiology. So, the liver, di ba, pag mag-study tayo ng anatomy, it is described as the largest gland in the body. So, it's a gland. It produces a fluid. And that fluid is bile. Bile is the excretory fluid of the liver. It is a micellar solution, which means meron kang ball of phospholipids, no? That are dispersed in water, in which the solutes are, so ito yung nagko-comprise ng ball of the micelle, bile acid, which is a modified cholesterol, phosphatidylcholine, which is a phospholipid, and cholesterol in a ratio of 10 to 3 to 1. So this ratio is important because each of these substances impart into the micelle a particular quality. The bile acids will again have both polar and non-polar groups. The phosphatidylcholine have polar and non-polar groups, and the cholesterol has a majority of non-polar groups. So for example, in surgery, we understand that if you change the ratio of these three, the solubility of your bile changes so that there is a propensity to produce cholesterol crystallization, no? Particularly when you have low levels of phosphatidylcholine in the bile. The cholesterol will separate from the two, and it will crystallize as cholesterol crystals, forming your gallstone. Hepatic bile is formed by active transport of the solutes by the bile salt export pump. Okay, so this occurs at the apex or the apical membrane of the hepatocytes. So ang ginagalaw ng liver cell would just be the solutes. And as it pumps the solute outside from intracellular into the bile canaliculi, habang tinatapon niya dun yung solute, magmo-move lang din passively yung water. And that is the process of bile formation. The bile canaliculi is formed by the apical surfaces of the hepatocytes that form tight junctions laterally. So may magandang picture dito si Dr. Net. Okay. Here, you will see the chemistry of the major bile acids. The high yield information here would be you should be able to differentiate the primary from the secondary bile acids. So again, all of them will stem from cholesterol. So the cholesterol will be acted upon by hydroxylases and dehydroxylase at carbon 27 to form cholic acid or the chenodeoxycholic acid. So the difference would be the hydroxyl group here. Okay, so I think this is carbon number 27. Ito. So the C27 dehydroxylase produces chenodeoxycholic acid. Okay. Next, this will be excreted by the liver through bile. But remember that bile flows through the rest of the gastrointestinal tract where it serves its function as an emulsifying agent for the fat so that it can be absorbed, but at the same time as a toxic substance for bacteria. But there are bacteria in the colon that continuously modifies the bile pigment or bile acid into the secondary bile acids. So yung primary bile acids gawa ng atay, yung secondary bile acids gawa ng colonic bacteria. So cholic acid becomes deoxycholic acid, nawala to. And chenodeoxycholic acid becomes lithocholic acid, nawala ulit to. And it also can become ursodeoxycholic acid. No? This in particular, ursodeoxycholic acid is utilized as an agent to solubilize gallstones, no? UDSA, ursodeoxycholic acid or UDCA. So the ursodeoxycholic acid is given as a capsule or tablet and then ma-absorb siya and it regenerates, no, or it adds up to the bile acid pool so that cholesterol will not crystallize out of the bile acid kasi mas dinamihan mo yung ratio. Sabi natin kanina may ratio na 10:3:1. So the cholesterol crystallization is because the first two, the bile acid and the phosphatidylcholine concentrations went down. So by giving UDCA, you return the ratio back to normal. So it prevents cholesterol crystallization. Okay. So the bile acids can further be conjugated by bacteria with either glycine or taurine. Okay. These are your amino acid conjugations at this position, the terminal carboxylic acid group. So again, this is an illustration from Dr. Netter. The primary acids are synthesized, conjugated, and secreted into canaliculi. In the gut, portion of bile acid is converted to secondary bile acids. However, the bile acids reabsorb into the portal system, that's your enterohepatic recycling, and return to the liver. In hepatocytes, the primary forms recycled and secondary acids epimerized and excreted. So this would be the blood side, and this would be the bile canaliculi side. So from the blood side, remember na uh, the portal venous blood would drain the small intestines, and the small intestines, in particularly the ileum, is where your bile acids are reabsorbed. So dito, kami ka ng bile acids reabsorbed. Um, it will reform the primary bile acids and then tatapon lang din ulit together with bicarbonate. So that means your bile is technically an alkaline fluid. Travels through the canaliculus and into the bile duct. So ito yung ating apical portion. Okay. So ganun din for secondary bile acids. But remember that the reabsorption of this is via secondary active transport. Ayan. So meron kang primary pump which removes sodium intracellularly, and the concentration of sodium extracellularly coupled with low sodium intracellularly is what powers the reabsorption. So that is your secondary active transport. Next, pinapakita lang dito yung inyong enterohepatic recycling. So ito, the new key information here would be there are a minority of the bile acids, around 5%, that are not reabsorbed by the ileum. And so they remain here in the large intestines where they are converted into secondary bile acids kasi nandito naman yung colonic flora mo. No? It becomes the secondary bile acids, and even the secondary bile acids are reabsorbed. But some are not reabsorbed, around 5%. And so you lose them through your poop. And so for extremely lipid-soluble drugs, no? Kahit paulit-ulit na siyang dumadaan dun sa phase 1 and phase 2, if it still is very lipid-soluble, it is eliminated through this route, through bile, and then some of it will not be reabsorbed, and so will be lost through poop. Okay? So, dito naman, pinapakita what happens when you have an obstruction. So, again, these are your conjugated bile acids already. The bile acids are not released. And so, the bile accumulates into the extrahepatic and intrahepatic biliary tree, and you lose bile that goes into the small intestines. What happens? Because the bile pigments causes the coloration of your poop, that's stercobilin, because it causes the poop to be brown if you don't have bile kasi na-block via obstruction. The poop will be ash colored. On the same note, the bile that stays in the biliary tree will reflux into the systemic circulation and find its way into the urine, where it will cause the urine to be tea-colored. So those are the signs that we ask, or symptoms that we ask a patient with probable biliary obstruction, colic stools, and tea-colored urine with jaundice. So this is what happens in cholestasis. The gallbladder serves to concentrate bile by actively reabsorbing the sodium ions. And again, when you reabsorb the sodium, the water just follows. And so you have a way of concentrating the bile. You can just imagine that the liver produces around 1 liter of bile per day, but the gallbladder only holds uh, 50, no? 50 ml of bile. So, paano yon? Paano nasa store ng gallbladder yung bile na ang dami-daming sinesekret ng liver? So, kinoconcentrate niya so that mas siksik yung bile solutes, no, that are present in the gallbladder bile. So, during fasting, the sphincter of Oddi, which is the sphincter that is uh, the circular smooth muscle that lines the ampulla of Vater. The sphincter of Oddi is tonically contracted, and so hepatic bile that is produced is redirected into the gallbladder. However, during feeding, the hormone released, which is cholecystokinin, causes the gallbladder smooth muscle wall to contract and the sphincter of Oddi to relax, which effectively releases bile to the small intestine so that it can solubilize the fat. So this is the picture, no? This would be your lumen side and this would be the blood side. So, ano lang din, secondary active transport lang din siya. Kukunin yung sodium dito and as it gets the sodium here, merong chloride na papasok, merong protons na lalabas. Okay? So, pag pumasok na yung sodium from here to here, the water will follow. And so what happens? The bile acids become more and more concentrated. However, they do not crystallize. Bakit? Kasi the decrease in water causes the bile acid solutes to form new micelles. By forming new micelles, hindi tumataas ang osmotic pressure. Okay? Kasi one micelle produces one osmotic particle. Okay? So, kahit na kumukonti yung sodium, kumukonti yung water, dapat tataas yung osmotic pressure, di ba? But no, hindi siya tumataas, isoosmotic pa rin siya with plasma. Kasi nagpo-form nga ng bagong micelles which produce only 1 osmol. And lastly, let's talk about how to assess the health of the liver even if we don't do a biopsy. So these are serum tests. For synthetic function, we test the serum proteins, and remember that majority of the proteins in the serum comes from the liver. So whatever that serum protein is, as long as you have excluded yung mga serum proteinopathies like amyloid, yung mga ganun, yung mga gammopathies from lymphoma, kung sure mo na walang ganun yung patient, then you can rely on the total serum protein as an indicator of liver function. Of course, pag bumaba siya, the liver function decreased. Another test would be just do your prothrombin time. So if you are able to measure the prothrombin time and the prothrombin time increases, ibig sabihin, yung mga coagulation factors bumababa. Bakit sila bumababa? Kasi nasisira yung liver cells. Okay, walang gumagawa sa kanila. Next, ALT and AST are intracellular enzymes. And so kapag nag-leak out sila into the plasma, that means sabog yung hepatocellular membrane. And of course, may sira siya. And kailan ba siya sumasabog? Only when there is necrosis. For biliary flow, you can test alkaline phosphatase and gamma glutamyl transaminase, no? So, these are specific, di naman very specific, but in the gastrointestinal tract, these are found in the mm, biliary epithelium. Okay? So, the biliary epithelium holds these enzymes. If tumataas yung pressure on the lumen of the biliary canal, the pressure is transmitted intracellularly and causes the biliary epithelium to release these enzymes into the bloodstream. Okay? So, when there is impaired biliary flow, for example, cholestasis, tataas itong dalawa. Okay? For detoxification function, you can measure the blood urea nitrogen, or more specifically, the ammonia. And for excretory function, you can look at the bilirubin and the bile salts. Okay, so here are my references for this lecture. Thank you for listening.